So what I'll be talking about today is a project that's ongoing in the lab that aims to address the question, does the blood-brain barrier contribute to pathological protein spread in Huntington's disease? First of all, we need to understand what is meant by pathological proteins and pathological protein spread. So this concept was first described in the 1990s by Stanley Prusiner in the context of prion diseases, and specifically with the prion protein. And prion diseases include things like Creutzfeldt-Jakob's disease and bovine spongiform encephalopathy. And what this idea states is essentially that a protein can be an infectious agent and can actually be the initiating factor in disease. Now, since the time of the description in prion disorders, similar mechanisms have now also been implicated in multiple neurodegenerative disorders, including amyloid beta and tau in Alzheimer's disease, alpha-synuclein in Parkinson's disease, TDP-43 and SOD1 in amyotrophic lateral sclerosis, and more recently and more controversially, mutant huntingtin in Huntington's disease. Now, mutant huntingtin is a particularly interesting case because Huntington's is a genetic neurodegenerative disorder where the mutant protein is already ubiquitously expressed. Now, before going further, I'm going to explain in more detail what it means for a protein to express prion-like behavior. And essentially, there are two mechanisms that a protein needs to convey to fit this description. So one, they need to be able to propagate from an area of relatively high concentration to other regions in the brain. And two, once there, they need to be capable of templating a change in the normal version of the same protein, such that the normal protein can take on a pathological conformation. Now, for the purposes of my talk, I'm going to focus a little bit more on propagation, although both of these processes are critical. So many different routes have been proposed for propagation. Some of them are just within the CNS and involve between cell types of the central nervous system, such as neuron to neuron by transsynaptic, but other processes are more general and can be between multiple different cell types of the central nervous system. So between microglia, neurons, astrocytes, and other cells that might be there. And this includes the release and then uptake of both free protein and protein contained within vesicles. The route that I'm going to focus on the most in my talk today is the possibility of transfer from more distal regions, such as the periphery, through the bloodstream. So this would involve the protein being in the bloodstream and moving into the central nervous system, or initiating in the central nervous system and moving into the periphery, which could be potentially a source of clearance of some of these pathological proteins. All right, so why are we interested in the blood-brain barrier and in circulation particularly? So this comes from an experiment that was performed recently in the lab where we performed parabiosis on a Huntington mouse to connect it to a wild-type animal. Now, because Huntington is a genetic disorder and the protein is ubiquitously expressed, it can be difficult to study spreading. But by controlling the source of mutant huntingtin within the wild type animal, we were able to evaluate if huntingtin was actually able to spread. And what we found is that after 6 months, we could actually see the mutant huntingtin protein within the brains of the wild type animals. So even though these animals do not express mutant huntingtin at all, we can see mutant huntingtin in their brains using two different methods of detection. So now if we come back to the schematic for a minute, you can see that one potential mechanism that we've drawn here is that this could be occurring via the LRP1 receptor. So why are we, and this is something that we are very interested in studying more. So why the LRP1 receptor? So our collaborators have done a lot of work looking at LRP1 in the context of Alzheimer's disease, and they've shown that LRP1 can contribute to the clearance of amyloid beta from the brain into the periphery through transport at the blood-brain barrier. And they showed this by using a knockout animal that doesn't express LRP1 in endothelial cells. And they saw that the clearance was greatly reduced in this context. There were multiple other transporters that could potentially be involved, because this didn't completely abrogate it, but it did suggest that LRP1 is involved in this. Now, in the context of Huntington's disease, preliminary data that we've generated with help from our collaborators suggests that LRP1 is in fact a transporter of Huntington, as can be seen here when we compared between LRP1 knockout mouse embryonic fibroblasts and mouse embryonic fibroblasts that contained wild-type LRP1. And there was a significant reduction of the amount of MHTT present within the cells that did not express LRP1. Now, this is all we knew when we first initiated this project, but a very interesting paper was also released just this year, which further implicates LRP1 in transsynaptic spreading of tau. So basically, tau was injected into the brains of mice, and then when shRNA targeting LRP1 was also added, you could see that the white tau protein was restricted just to the injection site and was no longer found throughout the brain. Now, this was particularly interesting to us because, as I mentioned, it can be difficult to study spreading of Huntington since you already have it everywhere. But if we can use a mechanism like LRP1 knockdown or knockout to prevent this transfer between different cell types, we can separate between the cell autonomous and non-cell autonomous toxicity of the Huntington protein. So after seeing this, we've modified our hypothesis a little bit. And so now we're interested in both understanding, so if LRP1 contributes to the clearance across the blood-brain barrier or the transport into the CNS from the periphery, but also if LRP1 transporter can contribute to disease progression, either through altering clearance or through modifying uptake and spreading within the central nervous system. So to answer this question and to address this hypothesis, we're working with two different models, an in vitro model and an in vivo model. I'm going to talk about the in vitro model first, which is something I've been developing for about the last year. So I started with human-derived induced pluripotent stem cells that come from just a control individual. And I've taken these cells and I've differentiated them into brain microvascular endothelial cells, astrocytes, and neurons. And then I've combined all of these cell types into a complex 3D model. And what I've been able to produce is I can actually generate a vessel that is completely impermeable to a 4 kilodalton dextran, and this is produced after about 5 days in culture. And we can have this endothelial cell in close proximity to cells of the central nervous system, such as astrocytes, pericytes, and even recently I managed to get neurons to survive in this perfusion system. To date, I've mostly been working on optimization, but we have managed to do a few preliminary experiments. We'll need to look at LRP1 in the future, but for now we did see that in our model, we can have that a fragment of the Huntington protein can cross from the endothelial cell compartment into the compartment with the central nervous system cells and the other direction. And we've done this both with mutant Huntington and wild type versions of the protein, and it does occur in both directions. Additionally, I looked at integrity of the vessel after exposure to these proteins and observed that there is a change in the permeability and that the barrier tends to become more permeable. This was particularly pronounced when it was outside the vessel and less so when the protein was placed inside the vessel. We have not been able to explore much into this, and this is still very preliminary data, but it's something that will be interesting to explore further and understand the role of LRP1 as these experiments progress. The second system is an in vivo system looking at a genetic cross between a Huntington mouse model, the ZQ175 mouse model, and an LRP1 knock-in model, which lacks the endocytosis functions of LRP1, but still allows the protein to be expressed and to go to the place it's normally found within the cell. So using this genetic model, we have generated this genetic cross, we've generated a number of different lines of mice, and we've begun very preliminary behavioral data. So this is only at 3 months, which is before onset of motor symptoms in Huntington's disease mice. But already at that age, we can see a modification in anxiety-like behaviors in both the changes in the amount of time spent in the open arms of the elevated plus maze, as well as time spent freezing in the periphery of the open field, which suggests that LRP1 may be involved in some aspects of disease progression. So briefly my conclusions, because I think I might be out of time. So we think that Huntington can cross the blood-brain barrier in our in vitro model, and that this transport is bidirectional, and that altering endocytosis functions of LRP1 can contribute and cause behavioral improvements in ZQ mice. So thank you to everyone that's contributed this project, and particularly to our collaborators in Germany. Questions? Oh, I can't hear anything. I think you're muted, Alice.
My bad. So I was just saying that audience, please post your questions in the Q&A section. That was a wonderful job, by the way. You stayed perfectly on time.
I just heard a beep at one point, and because I didn't have a clock I could see, so I'm glad to hear it was close.
You got it. You were perfectly on time. So we have one question. And
okay.
It says, is some stress or inflammatory situation promoting higher activity of LRP1?
So that's not something I've actually looked at yet, but it's a really interesting idea, and it's something that we'll need to look into more, because one of my theories about why we're seeing greater permeability in the blood-brain barrier is because there's an induced inflammatory reaction. So I think that's a really interesting idea, and I'd very much like to look into that more in the future, but I don't know right now, unfortunately.
Perfect. And then another question is, what do you think of the theory that Alzheimer's is also a prion disease?
So I think that prion-like mechanisms, oh, so do you mean that there is a problem with the prion protein specifically in Alzheimer's disease, or that there are prion-like mechanisms? So I think there is good evidence that the proteins such as tau and amyloid beta have prion-like mechanisms, and that can contribute to disease pathology. When it comes to the prion protein itself in the context of Alzheimer's disease, I'm not familiar with that literature, so I don't feel comfortable commenting on that.
Okay, perfect. And we have one last question. To what extent is LRP1 involved in transporting MHTT?
This is a question we're still trying to answer right now. So we've done a number of different experiments. I just showed one example where we did the knockout. We've also done some antibody inhibition. We're also working with multiple forms of the Huntington protein. So all I think we can say right now is it seems that LRP1 can transport Huntington, but it's not yet clear how much of Huntington's transport is dependent on LRP1 and which forms of Huntington can be transported of LRP1, but it's something that we're actively working on in the lab.
All right, so unfortunately, because of the time, we're gonna have to move on. There are a lot of questions, so please contact directly Melanie. So thank you, Melanie.
Thank you.
Now, reminder to all of the speakers and participants to keep your camera and microphone off unless you are presenting. And our next speaker is Mathieu. So Mathieu, go ahead. Excellent. So go ahead and share your screen.
Yeah.
Do you see the button? Perfect.
Is everything okay?
Could you speak a little louder? I think it might be a little bit difficult to hear you.
Is everything okay right now?
That's perfect.
Okay, thank you. All right, so I can start. So welcome everyone. So to start, I'd just like to ask, can trace amounts of contaminants present in your mother's body during pregnancy influence your brain's development? So the answer is pretty obvious for some contaminants. We have alcohol and tobacco that even though we're not exactly sure how and at what dose, we're generally concerned enough about it to say that it's better to be safe and sorry and not consume any. And this is an important point because these contaminants are generally consumed knowingly. Whereas for other contaminants, the answer isn't so obvious. So on this presentation, I'll touch about how prenatal exposure to legacy environmental contaminants influences frontal lobe integrity in adolescents. So what are legacy environmental contaminants? The name comes from because they've been well studied throughout history. The chemicals were commonly found in a lot of different appliances. So for lead, we can think about the use of leaded gasoline, leaded paint, and even water pipes here in Montreal. And there's two main classes of environmental contaminants. There's heavy metals and persistent organic pollutants. There's also the polychlorinated biphenyls, so PCBs, were commonly used as coolant fluids and flame retardants. But since the 1970s, most of these contaminants were banned for commercial use. And we can ask ourselves if they're called legacy environmental contaminants and their use has been phased out, haven't we examined all there is to know already? Well, no, because most of the known neurotoxic effects have been studied by postnatal exposure. And also, there's still some population that are more exposed today. So we can look at the Inuit population of Nunavik, where the levels that we measure in umbilical cord blood are 5 times more PCBs and 17 times more methyl mercury than their southern counterpart, like here in Montreal. And there's two main mechanisms behind this. The first one is the diet. So the principle of biomagnification is that the further you are up in the food chain, the more contaminants you accumulate because they're not evacuated from your body properly. So when you consume large marine animals like belugas and seals, you also consume a large amount of environmental contaminants. The second reason that this population is more exposed is because of their geographical location. So they're on the northern side of Quebec, and because of the atmospheric and oceanic currents, a lot of these legacy environmental contaminants are washed ashore. In terms of neuropsychological consequences, the literature find that mostly executive functions and functions closely associated to executive functions are affected. So for postnatal lead and methyl mercury, we can see that attention, cognitive flexibility, and working memory are affected. For PCBs, we see also response inhibition and set shifting deficits. And studies from the same cohort that I'm currently studying, so the Nunavik Child Development Study cohort, we saw that a follow-up of 11 months old and 11 years old, lead and PCBs were associated with attention deficits and symptoms of ADHD. Yeah, and we also know that the frontal lobe is very implicated in executive functions. So we had reason to believe that it might be affected by environmental contaminants in our cohort. And it's important to know because no study has investigated the link between prenatal exposure to environmental contaminants and frontal lobe integrity in adolescents. The Nunavik Child Development Study cohort started in about 1993 throughout 2002, where at birth we collected prenatal samples of environmental contaminants, so lead, methyl mercury, and BPCs in the umbilical cord blood. And about 18 years later, we flew 66 participants in Montreal to be assessed by MRI sessions, sociodemographic surveys, as well as postnatal blood samples, so current exposure to these contaminants. So the final sample that we have today is we have 66 participants aged about 18 years old and 60% women. The analysis that we did using the MRIs, we did some voxel-based morphometry, and we segmented some regions of interest, specifically the superior, middle, and inferior frontal gyri, as well as the anterior cingulate gyri, which we used to do some hierarchical regressions. The statistical model we built was in the block 1. We included the prenatal contaminant of interest, as well as confounding variables, so age, gender, education, drug and alcohol use of the adolescents. And in the block 2, we added the postnatal contaminant of interest. So there's two main reasons for the statistical model. The first one is that it enables us to see the relative contribution of both prenatal and current exposure on frontal lobe integrity, so on gray matter density. And the second reason is that it also enables us to see if the prenatal exposure currently influences the frontal lobe integrity, even when we control for current exposure. So we can look at the heavy metals results right now. We see that the prenatal exposure to lead predominantly influences both right and left superior frontal gyri. So just a quick recap over here. These are all beta coefficients of the prenatal variable of interest. And in the block 1, it's only the prenatal as well as the confounding variables. And in the block 2, we add the postnatal contaminant, but this is still the beta of the prenatal variable. So we can see that even when we control for postnatal, prenatal exposure to lead is associated negatively with gray matter density in the right and left. For methyl mercury, we can see that it's associated negatively for the right and left middle frontal gyri, even when we control for postnatal exposure. As for the PCB exposure, we can see that it mainly affects the left inferior frontal gyri, even when we control for postnatal exposure. It tends to stay significant. And in epidemiology studies, it's important to consider trends because we're looking at how a lot of different factors affect, a lot of different factors found in your environment influences you. So it's important not to overlook these normally not statistically significant results because they still are contributing factors. So these are all preliminary results. We looked at very large regions in the frontal lobe as an exploratory analysis. But we will be doing whole brain analysis that are currently on the way, and hopefully we'll be able to decipher more localized regions that are affected. And the next step, like Mr. Cizek talked during his speech, we would like to integrate neuroscience with psychology, and the way we want to do that is analyzing how environmental contaminants impact executive functions in our same cohort to see if the current link that we see between environmental contaminants and gray matter density loss mediates the executive functions. So what we can take from this presentation is that, first of all, legacy environmental contaminants affect multiple different regions implicated in executive functions. And these results suggest that they might have distinguishable impacts on certain executive function circuits. Also, all environmental contaminants are associated with loss of gray matter density. And the final and most important point is that it's important to take in consideration how during pregnancy, the food you intake and your geographical location, so your prenatal exposure, to even low-level doses of environmental contaminants, well, it can influence the frontal lobe's integrity even years down the road. Thank you.
Thank you, Matthew. That was excellent. So again, a reminder to the audience to post your questions in the Q&A section. And we have 2 questions for you already. The first one is, what did you use as a control group?
So we have no control group. Sadly, this is longitudinal studies. But we can look about because there's really not a lot of literature on this right now. The only other study that actually looked at influences of lead on the brain is in the Cincinnati cohort. So yeah, I'm afraid we don't have any control groups, but the results that we have specifically for the lead exposure, they're pretty much similar to what the Cincinnati group found a few years ago.
Okay. And the next question is, what are the routes of exposure you include as prenatal?
The routes of exposure, I'm not sure exactly I understand the question, but is it more like how people are exposed to prenatal contaminants.
I think so, exactly.
Well, the marine wildlife, the Inuit population consumes, like I said, like large marine animals, and we can find these contaminants in these marine animals. There's a lot of different levels of exposure. Like we use lead to hunt birds. We can be contaminated by this as well. And also the atmospheric currents, so in the dust that we find on the ground. That's it.
Perfect. And one more question is, how can you parse the differences between direct effects of the contaminants and possible effects that might be coming from the mother?
Yeah, this is a very good question. Unfortunately, since we only have 66 participants, it's tough to include all the variables we measured. But the variables that I included in the control group, other studies that were done on this cohort specifically said that these variables are important to include, like tobacco and alcohol, which are consumed by the mother, or affect frontal lobe integrity. But it's true to that other factors like anxiety in the mother also influences the development.
Perfect. That was excellent. Thank you so much again for your presentation. We'll move on to the next speaker.
Thank you.
And the next speaker is Laura. Go ahead and share your screen.
Okay, can you hear me?
Yes.
Perfect. Okay. Sorry, for some reason it doesn't want. Okay, can you see my screen?
Yes.
Okay. Okay, and my pointer. So, well, good afternoon, everyone. Thank you, Alice. And before starting, I would like to thank the organizers of this meeting for giving me the opportunity to present my PhD project, which aims to study the role of a kinase named pol-like kinase 2 in the pathogenesis and the treatment of Alzheimer's disease. So you've probably already heard about Alzheimer's disease. Briefly, it's Alzheimer's disease is the major cause of dementia that represents more than 50 million people all over the world, and unfortunately, these numbers are increasing every year. So Alzheimer's disease is an irreversible and progressive neurodegenerative disorder that is characterized by mainly by memory loss, but also other cognitive deficits. And these alterations are due to the presence of two proteinaceous aggregates in the brains of the patients. So on the one hand, we have the extracellular amyloid plaques that are mainly composed of amyloid beta or A-beta deposits. And A-beta is a peptide that results from the cleavage of a transmembrane protein named amyloid precursor protein, or APP. And on the other hand, we have the intracellular neurofibrillary tangles that are mainly composed of hyperphosphorylated tau, which is a microtubule-associated protein. And there are different factors that can modulate or can enhance the aggregation of these two proteins, notably the phosphorylation. Indeed, tau hyperphosphorylation has been largely characterized in Alzheimer's disease. It is known that the phosphorylation of tau by different kinases will prevent tau from binding to tubulin and other microtubule-associated proteins. And this will result in the destabilization of the microtubules, but it will also enhance tau aggregation into neurofibrillary tangles. And more recent studies are starting to focus on APP phosphorylation, and they suggest that the phosphorylation of the intracellular domain of APP can favor its cleavage through a non-amyloidogenic pathway or an amyloidogenic pathway. And this pathway can ultimately lead to the formation of the amyloid plaques. So all these events will be in part responsible for the neuronal dysfunction and death observed in Alzheimer's disease. And that's why it's really important to identify the natural kinases that will phosphorylate APP or tau, since the inhibition could represent a new therapeutic strategy for the treatment of Alzheimer's disease and related dementia. And well, in this context, our laboratory is interested in a kinase named pol-like kinase 2 or PLK2. So why PLK2? First, because some studies have reported a dramatic accumulation of PLK2 in the frontal cortex of patients suffering from Alzheimer's disease, as you can see by this twofold increase of PLK2 levels when compared to healthy controls. And other studies have suggested a direct interaction between PLK2 and APP. So PLK2 will phosphorylate APP, enhancing its amyloidogenic processing in cell culture. And collectively, this data suggests that the aberrant accumulation, but also the activity of PLK2 can enhance tau and APP pathology in Alzheimer's disease. So we hypothesize that PLK2 will phosphorylate tau and APP, enhancing the aggregation into amyloid plaques and neurofibrillary tangles, respectively, and thus that the pharmacological inhibition of PLK2 activity could reduce the toxicity associated to these two proteins. So the main goal of our research project is to evaluate the effect of the pharmacological inhibition of PLK2 in transgenic mouse model of Alzheimer's disease that presents both tau and APP pathology. And this is the triple transgenic mouse model. So this model presents A-beta and tau pathology and also cognitive deficits. And what we did was treat wild type and triple transgenic, both males and females, for a month at the initiation of the pathology. And during this month, the animals receive a daily intraperitoneal injection of a specific PLK2 inhibitor named compound 37 or saline as a control. And 2 months after the treatment, when at an age in which the pathology is completely established, we perform several behavioral tests and we sacrifice the animals for further immunohistological and biochemical analysis. And now I'm going to show some of the results obtained at the behavioral level first. So we wanted to evaluate the effect of the treatment in short-term recognition memory. And for that, we use the novel object recognition memory. So briefly, the animal is allowed to freely explore 2 identical copies of the same object. And 1 hour later, one of the familiar objects will be replaced by a different one, referred as the novel object. And we expect healthy animals to spend more time exploring the novel rather than the familiar object. That's why we calculate the discrimination ratio between the novel and the familiar, as you can see in this graph. And indeed, wild-type animals that you can see in green spend more time exploring the novel than the familiar object, showing good levels of recognition memory. However, this discrimination ratio was affected in saline triple transgenic mice, so non-treated triple transgenic mice, reflecting a deficit in short-term recognition memory. And interestingly, the treatment prevented this, or partially prevented this effect, as you can see by this amelioration of the performance in orange for triple transgenic mice that received the PLK2 inhibitor. And after the behavioral test, we collected the brains, and first we evaluated the effect of the treatment on the amyloid plaques in triple transgenic mice. So for that, we use Congo red staining, as you can see in the image, and we quantify the number and the surface of the plaques in the hippocampus. And as you can see, we did not observe significant differences in the males, neither for the number of plaques or the area. However, for the females, although we did not see an effect of the treatment in the number of the plaques, we saw that the pharmacological inhibition of PLK2 decreased the surface occupied by these plaques in the hippocampus of the females. And in parallel, we quantify some synaptic markers. So I'm showing a couple of them here in the forebrain of triple transgenic mice by Western blot. And as you can see, there was an increase of PSD95 and synaptophysin in treated males in orange when compared to the saline group that is represented in blue. And for the females, we did not see a difference between non-treated and treated mice. So PLK2 pharmacological inhibition seemed to increase different synaptic marker levels in the forebrain of male triple transgenic mice. So to sum up, we hypothesized that APP, well, PLK2 will phosphorylate APP. This will enhance its amyloidogenic processing, the accumulation and aggregation of A-beta into amyloid plaques. And so far, we've seen that the pharmacological inhibition of PLK2 prevented or partially prevented the memory deficits observed in this model. We also saw a decrease of the surface occupied by the amyloid plaques in the hippocampus of triple transgenic females. And finally, an increase of the synaptic marker levels for the triple transgenic males. So overall, the pharmacological inhibition of PLK2 seems to ameliorate or prevent some of the neuropathological aspects in this transgenic mouse model of Alzheimer's disease. And we also hypothesized that somehow, either directly or indirectly, PLK2 enhanced tau hyperphosphorylation and its aggregation into neurofibrillary tangles. And that's what we are currently exploring right now. Like, did the treatment had an effect on tau pathology? And this could maybe explain the differences that we see on the effect of the treatment between males and females, for example. So this is what we are currently working on. And well, to finish, I would like to thank my supervisors, Dr. Weslati and Dr. Rivet and their teams, as well as our collaborators and the funding organizations. And thank you very much for your attention. Don't hesitate if you have any questions. Thank you.
Thank you, Laura. That was amazing. Now we have slide up. I'll try to get to it as many as possible, but if I don't get to your question, please, at the tables, you will be able to ask the speaker some more questions. So for now, the first question is, at what stage of AD does the PLK2 levels increase?
Okay, so in humans, it has only been studied in advanced Alzheimer's disease stage. So I cannot tell at which time point, let's say, this increase will happen. We assume it will be gradual. And there is another study done in mice that shows spatiotemporal increase of PLK2. So basically, over the time, PLK2 levels will increase. It was they did that in a similar model than the triple transgenic, but that just show APP pathology. But it's also like an aging model. So when they are old, they will have a progressive increase of PLK2, and this will be region specific, and it will switch over time.
Mm-hmm. Perfect. And there's one question that's asking, what would be the reason for you, or like, would you choose to treat the animals before the plaques appear?
Well, yeah, that's something we are currently working on. So I would say the problem of the model we use is that there is a difference in the establishment of the pathology between males and females. The pathology will start earlier in the females and will be more severe. So we compromise something in between. But I'm currently also working in a faster model that is the APPPS1 model. And for that, yes, we are treating before the appearance of the plaques to see if we can prevent the appearance, if that was the question.
Oh, amazing. Awesome. Okay, we have time for one more question. Okay. So it says, in both your 3X TG and your 3-XTG treated with your compound, your data is clustering roughly into 2 distinct groups. How would you interpret this?
Okay, so yeah, that it's like one of the problems we encounter is the variability between the mice, especially at the behavioral level. What we can read out of our data, also data that unfortunately I couldn't show here, is that we have 2 populations. So some of the non-treated triple transgenic will not show deficits, or not, the pathology will not be as severe as we expect for the age they have. But in general, for the treated mice, we don't have populations that show severe, strong deficits. So we are aware of this problem, and it will be actually interesting to increase our N, to add more controls to the experiment to answer actually this question.
Awesome. Thank you so much for the wonderful talk. We're going to move on to our next speaker. So again, attention to the audience. If you have some more questions left, you will be able to ask them on the floor at the speakers table. So right now we're going to move on to our next speaker, Azalée. So Laura, go ahead and stop your screen share. Perfect.
Hi, can you hear me?
Awesome. And hey, yeah, I can hear you. Go ahead and share your screen.
Okay, here. There you go. Technical difficulties on. Okay.
Perfect.
So, hi everybody. My name is Azalée Mongrain, and I'll be presenting our study on the influence of emerging literacy on brain activity of the reading network. So, as we all know, reading is a process that allows us to give meaning to written code, and this process is necessary in order to acquire knowledge, since it allows us to access a wide variety of information. This is one of the reasons that literacy skills are predictors of academic success later on. More specifically, as opposed to speaking, reading requires explicit training and can't be learned simply by imitation. And this is probably due to the fact that reading is a relatively recent cultural invention dating back to around 5,400 years ago. This implies that there's no brain region specifically dedicated to reading in itself, and that any cortical specialization is actually due to the brain's ability to reorganize itself with its experience. So thanks to advances in neuroimagery, we do have an idea of what goes on in an adult reader's brain. So reading is a complex process and involves many pathways in different regions. One of the regions that is consistently linked to word and letter recognition is the visual word form area, located in the junction of the occipital-temporal cortex in the left hemisphere for the majority of readers. And this visual word form area acts as a bridge between visual stimuli and language processing that allows us to recognize words and letters during reading. The visual word form area is actually the specialization of the visual word form area depends on experience, and our literacy skills actually correlate with the visual word form area's activation. So we do know what goes on in the brain during an adult's reading process, but we were wondering what signature neural activity can be detected in children during emerging literacy. So research generally shows a visual word form activation after at least 1 to 1.5 years of formal education. And this implies that a relatively high amount of literacy skills is required in order to start the lateralization process that we typically see in adult readers. However, one recent study by Lashy and al. Found a left occipitotemporal activation in 5-year-old pre-readers exposed to written code using a different EEG approach, more adapted to children having smaller testing periods and a higher signal-to-noise ratio. So Lashy and al.'s studies actually suggest that basic literacy skills suffice to start the lateralization process typically seen in adult readers. So that being said, we began to ask ourselves, so what is actually going on during emerging literacy? Can we find occipitotemporal activity in readers with basic literacy skills? And can those basic literacy skills be correlated with visual word form area activation? In order to answer this, we originally wanted to do a study on 80 children from the ages of 4 to 8, where we wanted to assess brain lateralization related to the processing of written words in pre-readers and readers. However, due to the current situation, we were only able to do a pilot study on 5 adults, where our goals were to validate our experimental protocol for the original study. We were expecting a visual word form area response in our adult participants exposed to written code. We were also expecting that words and pseudo-words would activate the visual word form area in a similar way, pseudo-words being an arrangement of letters that make grammatical sense but that don't make semantic sense. So in our original study, we had many tests assessing basic literacy skills and cognitive functioning, whereas in our pilot study, we only administered the EEG protocol that was based on Lushy and Al's method. So here we have, I'm not even in Jumo. So here we have our EEG protocol that was based on 3 conditions in which oddball stimuli were periodically inserted in a series of base stimuli. Our first condition being words inserted in a series of pseudo-fonts. Our second condition being pseudo-words inserted in a series of pseudo-fonts. And our third condition being words inserted in pseudo-words. So at the bottom of the screen here, we can see an example of the words pseudo-font situation that was played during our testing. So each condition lasted 40 seconds, was repeated twice at a rate of 6 items per second, with an oddball appearing every 5 base stimuli. And all stimuli were based on 4-letter French words taken from a Quebec database. So here we have the grand average topographies per condition for the oddball responses. We have the fundamental frequency here at 1.2 hertz that we see on the top. And for all 3 conditions, we can't see a clear left lateralized response. This is actually not surprising since it's a relatively low frequency and it's more noisy. Whereas when we look at the harmonics, 2.4 and 3.6 hertz, we do see a left cortical activation for the pseudo-word pseudo-font condition and the word pseudo-font condition. Both these conditions are activated in a similar way and contrast with the last condition, the words inserted in pseudo-words condition. The fact that we don't see activation in the words pseudo-word condition actually relates to our hypothesis that words and pseudo-words activate the visual word form area in the same way. We can also see our base response here at 6 hertz. We have a bilateral occipital activation, which isn't surprising since our stimuli are visual stimuli. Here we have the same results, but represented on an EEG spectra. So we can see here the fundamental frequency. It's difficult to see the activation peaks, whereas on the harmonics here, 2.4 and 3.6, we can clearly see the activation peaks for the first 2 conditions. There. Here we're presenting our grand average signal to noise ratios per condition for the O1 electrode, which is the electrode we use to assess the visual word form area. We can see that the pseudo words and words inserted in pseudo fonts conditions, the 2 first ones, have similar signal to noise ratio values, and that these values are higher than the words to pseudo words signal to noise ratio values. This relates to our hypothesis, once again, that words and pseudo-words activate the visual word form area in a similar way. Furthermore, we can see that for the first 2 conditions, our SNR values are higher than the SNR values for the O2 electrode, which is the corresponding electrode in the right hemisphere, suggesting that the activity we see is specific to the left occipitotemporal region. So to conclude, we have confirmed our hypothesis. We did find a visual word form area activation in our adult participants. We failed to find an activation difference in pseudo-words and words, which is actually consistent with the literature, where many studies don't find a difference in activation of the visual word form area for words or pseudo-words. This might be due to the fact that a visual word form area might be involved before semantic processing. Our pilot study had a few limits, the first one being our small effect size with obviously 5 participants. The second one being we did it on adult participants. So the protocol was based for children, but we tested it on adults. Adults have thicker scalps. They have more mature reading processes. And so our future goals are to complete our original study, to extend the protocol study on children, first of all, and in hopes of assessing the evolution of the specialization of language regions in pre-readers and in readers. And eventually, we would also like to assess autistic hyperlexic children with the same protocol in order to bring more comprehension regarding atypical models of reading acquisition. So thank you. I would like to thank Alexia, Chloe, Laurent, and Dave for working on this project with me. I'll be here if you have any questions.
Perfect. Thank you so much. That was excellent. We have one of the questions is asking, does visual word form activation relate to skill in children, or do some skilled readers have higher degrees of activation?
Well, the study we're basing our study on actually measured that. They tested for literacy skills in children, and they did see that the literacy skills correlated with the visual word activity. They also assessed basic literacy skills, so they do correlate with visual word form activity. Part of our original study is replicating those results, since it's the first time we've had results like that on 5-year-old children. So we're hoping to confirm that hypothesis once again. Yeah.
Perfect. And we unfortunately only have time for one question. I apologize, the other audience. So the question is, do you think the amplitude of the VWFA will scale with how easy the letters are perceived? For example, replacing the pseudo words with words with distorted letters.
Okay, well, I know that the visual word form activation is uniformized. I don't know how to say that in English, but no matter what the invariable, no matter how the word is presented, the activation is the same for the same letters. So if you have a capital letter or a non-capital letter, the activation will be the same. And pseudo words and words, we've already seen that there's no real difference in the amplitude of VWFA activation. Okay, I'm just going to make sure I answered all those components. Yeah. Also, what's interesting is that we took different levels of words. All our words were four letters, obviously. But since we have a wide variety of children from 4 to 8 years old, we couldn't take only easy words and only difficult words. And we're probably going to be controlling for the level of frequency of exposure in the environment. So we might have maybe exploratory results regarding that question later on. Yeah.
Awesome. Thank you so much for your presentation. We're going to have to move on, unfortunately, due to the time restriction. So our last speaker for this session of the oral session is Adam. So Adam, go ahead and share your screen. Turn on your mic as well.
Thank you for having me.
No problem.
Okay.
Great. So make sure to put your presentation on.
You can see it?
There you go.
Okay. So my project is titled The Dark Side of Heme Oxygenase 1, or HO1. So the Shipper Lab at the Lady Davis Institute of the Jewish General Hospital has over the past 30 years published over 200 peer-reviewed papers on oxidative stress, brain aging, and neurodegenerative diseases. The overarching objective of the lab is to ascertain the roles of heme oxygenase 1, iron-containing astroglia, and oxidative mitochondrial injury in Parkinson's disease and other aging-related neurodegenerative disorders. There's a substantial body of evidence attesting to the beneficial roles of HO1 when it's upregulated in the brain. However, in some cell types and under certain conditions, HO1 may amplify intracellular oxidative stress and exacerbate the disease process. And this inimical side of HO1 is often ignored, which I've mentioned, and it is the subject of this presentation. Parkinson's disease is the second most common neurodegenerative condition. James Parkinson described Parkinson's disease. He was the first one and called it a disease of long duration with involuntary tremulous motion, lessened muscular power, and a propensity to bend the trunk forward. The hallmarks of Parkinson's disease are, firstly, the degeneration of dopaminergic neurons in the substantia nigra pars compacta. Secondly, there is formation of Lewy bodies in various brain regions. The risk factors for Parkinson's disease include a complex interplay between hereditary factors, sex, environmental, and occupational exposures. Dopamine replacement therapy, which is the most commonly prescribed treatment for Parkinson's disease and consists of levodopa, which is a dopamine precursor, along with carbidopa, a peripheral dopamine decarboxylase inhibitor. And these treatments fail to slow or stop the pathogenesis of Parkinson's disease. Heme oxygenases are part of the oxidoreductase family, which in combination with NADPH and cytochrome P450 reductase catalyze the degradation of heme into biliverdin 9A while releasing free ferrous iron and carbon monoxide in all mammalian cells. Biliverdin 9A is then reduced by biliverdin reductase and NADPH to bilirubin. Heme oxygenase 1 expression is confined to a few glial cells and some neurons, while heme oxygenase 2 is ubiquitously expressed in the central nervous system. Pro-oxidant, free ferrous iron, and carbon monoxide are produced during heme degradation, while antioxidant, bilirubin, and biliverdin are also produced. And heme oxygenase 1 expression can be induced by many factors, such as dopamine, tumor necrosis factor alpha, and interleukin-1 beta. A considerable body of evidence adduced by our laboratory over the last 3 decades implicates glial HO1 in the pathogenesis of Parkinson's disease. Firstly, we found that HO1 is more abundant in the neuromelanin-containing substantia nigra pars compacta neurons compared with controls in Parkinson's disease postmortem brains. In addition, the Lewy bodies in neuromelanin-containing cells of postmortem PD brains contain high levels of HO1 protein. Secondly, transfection of rat primary astrocytes with the human HO1 sequence results in a core neuropathology, which consists of pathological iron deposition, oxidative stress, mitochondrial damage, and macroautophagy. Third, ferrous iron derived from the degradation of heme is capable of converting dopamine to neurotoxic compounds in HO1 transfected primary astrocytes. And fourth, the chronic HO1 overexpressing mouse model demonstrated that glial HO1 is a driver of PD pathology in mice. Although exceptions exist, the behavior of HO1 in the setting of acute neurological illness, such as stroke or traumatic brain injury, is more often than not trophic in nature. In contrast, the participation of HO1, of neural HO1 in chronic states, such as central nervous system senescence, Parkinson's disease, and schizophrenia, largely by facilitating the accumulation of redox active iron, may be predominantly maladaptive. Based on this, we have formulated the hypothesis that astroglial HO1 expression is neuroprotective in the acute phase of exposure to Parkinsonian toxins, but may exacerbate toxicity in chronic phases of the illness. The ALDH1L1.HMOX1 knockout mouse, which I'll refer to as the transgenic mouse, expresses a fusion of Cre recombinase and mutated ligand binding domain of the estrogen receptor, ERT2, under the control of the human ALDH1L1 promoter. When the latter are crossed with a strain containing a LOXP site flanked sequence of interest, Cre-mediated recombination results in deletion of the target, that is tissue-specific. The Cre-ERT2 strain is conditional because CRE-ERT2 is retained in the cytoplasm unless stimulated by the estrogen receptor antagonist tamoxifen to translocate to the nucleus and induce targeted mutation. The transgenic mice remained unexposed to tamoxifen until 2 months of age. At 3 months of age, the transgenic mice and the wild-type mice received the dopamine neurotoxin rotenone. Rotenone was administered at either 1.25 or 2.5 milligrams per kilogram for 35 doses over a period of 35 days. 2 weeks later, behavioral, neurochemical, and neuropathological endpoints were surveyed. Males and females were studied. In the first 3 days of treatment with 2.5 milligrams per kilogram rotenone, 2 transgenic mice were found dead in their cages. Then, to avoid loss of more transgenic mice, which were novel and just generated, the dose was reduced to 1.25 milligrams per kilogram. On day 9 and 10 of rotenone treatment, 2 wild-type mice were found to be lethargic, unresponsive, and with respiratory difficulty, and so they were sacrificed. Then on day 15, another wild-type mice was found in poor condition, and he was sacrificed. Mice were weighed on a weekly basis, and on week 3 of treatment, many mice, including mice treated with vehicle or the rotenone treatment, developed lumps around the injection site and appeared thin. The identification of locomotor abnormalities in PD models requires the use of tests which assess motor coordination, balance, and general locomotor activity. PD models manifest locomotor abnormalities to varying degrees. In addition, other behavioral tests can assess anxiety and olfaction in mice. In order to assess locomotor abnormalities, the footprint test, the buried pellet test, the pole test, grip strength test, and general locomotor activity were monitored. In the open field test, the forepaw hindpaw analysis, and the grip strength test, there was no significant interaction or main effects. However, in the pole test, there was a significant main effect between rotenone-treated and vehicle-treated mice. Impaired olfaction appears as a symptom in prodromal PD. The buried pellet test was used to assess impaired olfaction. However, there was no significant main effects. And anxiety was also assessed because anxiety can be a confounding factor in behavioral experiments. And so it was measured using the Thatcher-Brighton paradigm. And this was also non-significant, suggesting no anxiety there. So based on our findings, we conclude that astragalil HO1 may be neuroprotective in the early stages of exposure to rotenone, but may exacerbate rotenone toxicity in the later stages of the model. In addition, no significant protection was conferred to the transgenic mice on the basis of the locomotor behavioral testing, and tyrosine hydroxylase staining in the substantia nigra and striatum should be performed. In a future study with MPTP, we predict the transgenic mice will be protected from toxicity. And since I'm running out of time, I just like to wrap it up quickly by saying that there's a strong need to develop neuroprotective treatments for Parkinson's disease, and the data that our lab has generated over the past many decades implicates HO1 as a target and recognize it as a target to treat idiopathic Parkinson's disease. Thank you.
Thank you so much, Adam, for being super considerate. Let's see if we have time, And in the meantime, I want to say thank you to the audience. We had over 100 viewers watching live. That was incredible. Okay, so we have one question. Is the death typical of the transgenic mice?
No, it was actually very well. It's the first time this transgenic mouse has been generated, so it's completely novel. This was the first test with a Parkinsonian toxin as well, so it was unclear at first why this happened or what caused the death, which is something we're actually particularly interested in finding out. But as I mentioned, actually, since the role of HO1 we've kind of shown in our lab that it is in the case of acute illness, such as acute exposure, like to a Parkinsonian toxin in the first 3 days, is possibly protective and trophic in nature. So by degrading heme, it's protecting the brain. However, in chronic phases, it's more detrimental towards the brain and to the dopaminergic neurons.
Gotcha.
So.
Awesome. Thank you so much, Adam. All right, so now we're going to, oh, so by the way, to the audience, please don't forget to vote. So the voting is going to be open for just a few minutes for this particular oral session. So go ahead and vote right now for the presentations that you enjoyed. And the link is going to be posted in the chat. And we are going to switch gears right now and move on to the next session. So thank you, Adam.
Thank you. Thank you. Bye.
Bye.